communicate between one another, how cells sort of communicate within one another. And then finally, we're going to sort of talk about how cells replicate, how we make gametes, which are called sex cells. And from there, I'm going to start to talk next class about a little bit more in-depth things about, again, staying in molecular biology. So just as a note, for those of you that have done assignments, or you go and you don't have a grade for an assignment, you should look to see if you've turned it in. So there's a number of people that have not actually turned in the assignment that they were working on for, well, for weekly number 1, it's a bit too late at this point, but for weekly number 2, there's some people that are still working on the exam, I noticed. But you should just make sure you turn it in, just because if you don't turn it in, I can't actually give you a letter grade for it. So just make sure you turn it in so you can get graded for it. And remember, getting even partial credit is better than getting no credit at all. So, okay, with that out of the way, let's get to talking about cells. And so just as a sort of refresher, we talked about generalities of the plant and animal cell, and there are two very similar concepts, but they do have overall different structure. Remember, plant cells have cell wall, they have a vacuole, whereas our animal cells, only thing that differentiates them from plant cells is the presence of a centriole, which is this little thing here. Now remember, eukaryotic cells are specialized. Everything they do is done in some sort of compartment. So, and these compartments are generally what we call organelles, or organelles being mini organs. And remember, we went through sort of all these different organelles: the nucleus, where we store DNA; the nucleolus, where we have a stress response and the production of ribosomes, which again, ribosomes making proteins. We talked about DNA previously, DNA being stored in chromosomes. This is the basic information of your cell. The rough and smooth endoplasmic reticulum, which are the site of protein synthesis as well as lipid synthesis. We have our our lysosomes, which is the site of recycling in your body, or at least in your cells. We have the mitochondria, which is the powerhouse of the cell. We have the outer membrane, which again separates self from non-self. And finally, we have the Golgi apparatus, which is something that packages and ships things all around your cell. There's one other thing we didn't talk about, which is the perioxisome. Not a super duper important organelle, but it does have a similar function to the lysosome. And so one of the key things we didn't really discuss last class is actually the cytoskeleton. And we're going to talk about this today because it plays an important role for sort of how things move within inside of a cell. It sort of has this little double-edged function where it's both structural and important for transport within inside the cell. But as I sort of mentioned before, one of the things that is important about biology and chemistry is that structure and function are linked together. So depending on the structure you have, that will dictate what function you can do. That's true for chemistry, that's true for evolutionary things like traits. This is also true at the cellular level. And just to sort of illustrate what I mean by that, we can look at a couple different classes of cells here. So first we can look at a pancreatic cell. For those of you that don't know what a pancreas does, it produces, helps in digestion. It also seems to be a site where we keep good bacteria. That's some of the newest research about your pancreas, which I think is pretty cool. But we keep lots of digestive enzymes. And you'll notice that it's dominated by rough endoplasmic reticulum. And if you remember, rough endoplasmic reticulum are the place where we make proteins. So we're making enzymes, which are just a specific type of protein. And and you can look at cells inside of your pancreatic cell are chock full of these rough endoplasmic reticulum. That's in contrast to, say, something like a plant leaf, which, as we talked about, has a huge vacuole. So it has lots of capacity to store water, which keeps the plant full of turgor pressure, which, remember, keeps a plant stiff and upright and rigid. In addition, plants, again, will have many, many, many chloroplasts. Sometimes plant cells will have hundreds of chloroplasts. Sometimes you only have a few. Inside the animal testicles, they have lots of smooth endoplasmic reticulum, because one of the main components that testicles do produce are lipid-soluble signals, or just fats. including hormones and steroids. And so you have lots of smooth endoplasmic reticulum because that's the site of fat and lipid synthesis. So, and then finally, we have what are called brown fat cells. These aren't particularly very common in adult individuals, but babies have lots of brown fat. And for those of you that have never heard of what's called a brown fat cell, they basically are basically like chunks of like your skin. You technically have some on the back of your neck, which is why wearing a scarf is really good, because what brown fat cells actually do, and they do adults, they do in babies, is they produce a lot of heat. So what they end up doing is they burn fat to generate heat instead of ATP. So these cells help keep warm. So for those of you that have adopted a baby, like infant size, you would notice that infants never shiver. And one of the reasons they never shiver is because they're chock full of this brown fat. And one of the reasons you can wear a scarf and it has a really disproportionately large effect on keeping you warm is because of the brown fat cells, which you have still in the back of your neck and in your neck as well. So, but you can see that they have lots of mitochondria because they're trying to take energy. They're trying to make energy, and the energy they're trying to make is heat. So in terms of repeating what the function of brown fat is simply just to make heat. So it's just a special type of cell inside your body. Again, it's really, really rare in adults. Again, you really only find it on the back of your neck. But in babies, they're covered in brown fat cells, and they sort of mature away and go away as you get older. But brown fat cells basically just take all the energy, in particular, they take fats that you're consuming or fats that are part of your body, and they essentially stick those in the mitochondria. And instead of trying to generate lots of ATP, they actually just try to generate heat. And so as we've talked about, one of the components of these chemical reactions your body is doing is waste energy, right? No energy is wasted inside of a system. And one of the things you can do as you're performing cellular respiration inside of your mitochondria is produce heat. So all the heat that makes you your steady 98.6 degrees, or plus or minus a degree, all comes from your mitochondria. So your sort of consistent regeneration of heat inside your body is all from your mitochondria. Brown fat cells just do this, and they essentially skip making ATP, and they simply just try to make as much heat as they physically can. And all this heat is good for babies because babies need to be warm. Babies also don't shiver. So that's brown fat. It's actually a kind of interesting thing. I actually only learned about brown fat a couple of years ago from my friend who had a baby, and he was all into baby biology. So. Anyways, so this is the sort of the specialization you can see at the cellular level. Now, remember, bacteria don't quite do this. Their specialization that they do is really at the genetic level. They just change their genomes. They acquire new genes. They don't have organelles, so they don't do any of this level of specialization. The specialization they're doing is metabolic, not cellular component-wise. So we're going to talk about how we put all this together, how we sort of go from these changes, how we go from all these organelles together, how we put it together to make a cell work. And again, as I mentioned, we're going to have to talk about the cytoskeleton. And this is basically a series of tubules made out of proteins and other carbohydrates that basically give your cell the structure. They also are used for movement, so they're allowing your cells to move, even if it's just slightly. And they also anchor your organelles, so they're not just sort of kind of everywhere. I think one of the things I may have neglected to mention about your cell is all these organelles are sitting in what's called the cytoplasm. So your cells are basically, as I mentioned, just a big bag of water. And the cytoplasm, again, is just basically a big bag of water. And it's sort of all the dead space inside your cells that is not filled with organelles. And your cells, if they weren't anchored, your organelles, if they weren't anchored to something, they would just be floating around in this sort of ill-defined liquidy substance. And so to keep them sort of anchored in the same spot, as you can sort of see from this graphic cartoon here, is they're anchored by what's called the cytoskeleton here. And so the cytoskeleton is going to be sort of our first topic of the day. And this is sort of what a cytoskeleton looks like. So these are muscle cells. And muscle cells, they take all these sort of weird and funky shapes. They're not that standard circular shape I've been showing you. But I think that's pretty intuitive to you guys. You know, muscle cells have to be flexible. They have to move, and muscle cells can be long, right? I mean, I'm sure you've probably always seen either cartoonized versions or potentially like had the potential to dissect some sort of animal and seen what muscle tissue looks like. And it's long, it's fibery. And when we look at sort of a piece, sort of this microscopic view of a muscle cell, you'll see that we have the nucleus. It's stained in green here. And we have two components of the cytoskeleton here. We have the microtubules, and these are long, sort of spindly-like things. They sort of anchor, you can see they anchor the nucleus, and they sort of spread out through the entire sort of network of the cell. And then you have sort of this underlying component stained here in blue, which is called the actin. And many of you probably have heard of actin, you probably have heard of pectin. There are all sorts of components of the cytoskeleton. So this is what the cytoskeleton looks like. Again, it provides an anchor and it provides an overall structural support for the cell. Because remember, like when you take a step, right, you're stepping on your cells, right? And that cytoskeleton gives that sort of cells the overall strength to say step on them. But in terms of presence and absence, when we're thinking about the cytoskeleton, actin is not a particularly very common thing. It's generally only found in animals. It's more of a muscle tissue sort of piece of the cytoskeleton. Microtubules are pretty much the most common thing found in animal cells as well. What do you mean by the chemical formula / buildup of muscle spasms?
So I mean, like, you know how you like have a structure for a carbohydrate? Like, I mean, the proteins. Like, if you could, are we supposed to know like what a protein is supposed to look like when it's all built up, like all the chains, you know, like in a lipid cell? Like we had to, I think we looked at that. Like, is that going to be on the test or anything?
No, I mean the test is already available for you to take,
Okay.
so you can go see exactly what's on the test.
Yeah.
No, there's no specifics like that. I'm not that kind of professor. So if you have any, the test is there. If you have any questions, just let me know. But the sort of specifics are sort of beyond the point of this course. If you guys were actual biology majors or chemistry majors, specifics are much more important. But now it's more sort of broad topics. That's what.
Okay, cool. Thanks.
Okay, so moving on, cytoskeleton structure, but as I mentioned, it also provides a network for movement and sort of provides this anchoring for all your organelles. But the network for movement is really sort of one of the more important things for today's class. Is while this does provide, again, some structural support and so on and so forth, the key here is how we use these tubules for movement. So we're going to start by talking with the nucleus, because the nucleus, as you all know, is the brain of the cell. It's where we store our DNA. The nucleus also has the nucleolus, where we manufacture ribosomes, which our ribosomes are then where we make proteins, right? So that's the sort of flow of information in a cell, right? DNA is transcribed to RNA inside the nucleus, and then that RNA is exported out of the nucleus to the ribosomes, and the ribosomes, remember, are synthesized by the nucleolus. Now, remember the message in terms of the overall storage of information in every cell, bacteria, archaea, eukaryotic, is DNA. And we go through the central dogma of biology, DNA to RNA to protein, to do action. So remember, you transcribe your DNA to RNA, and then you translate it in the ribosome to an action. And that's sort of the flow of information, the instructions to do everything a cell does. You're trying to make a lipid for you're trying to make an enzyme to digest things. It has to go through this pathway. Try to make some lipids. It has to go through this pathway. You're trying to respond to, say, a bacterial infection. It has to go through this specific pathway, which I think is nice. It makes things more simple to understand because it's just a one sort of street pathway from information to action. But it also means that you can't really do anything that's not on this pathway. You can't you can't just do something that your DNA doesn't encode. You can sort of subvert that at the sort of the human level by taking medications, right? You can, if you have a headache and you don't have quite your instructions don't let you sort of ease your headache. You can take medication that works with your cells to ease your headache, but you might not have the instructions to ease that sort of headache. That was a bad example, but that's the sort of idea that we're going with here. So again, but it all starts with the nucleus, this cool dude. And so this is sort of the pathways we're working in. It's called the endomembrane system. And again, it all starts with that nucleus. So we have to take that DNA, transcribe it to RNA, export it out of the nucleus. And now where does that RNA go? Where does it find its ribosomes? Well, it finds them in one or two places. You can see that here, sort of in this graphic image, you can see there's ribosomes kind of hanging out in the nucleus. I'm sorry, the cytoplasm here, that sort of empty space. That's not particularly common. The most common place your RNA is exported to in your cell is the rough endoplasmic reticulum. And remember, this is the main site for protein synthesis. Now, once you send it here, it's essentially the ribosomal RNA. I'm sorry, the mRNA is sent to one of the ribosomes here, and you transcribe it. I'm sorry, you translate it into proteins. Now, once you make that protein, you do one of two things with it. You either stick it in a vesicle, so basically you take a piece of this rough endoplasmic reticulum membrane, you bud it off, and you send it to one of two places. You either send it to the smooth endoplasmic reticulum, where that protein is then used to make a lipid, or you send it directly to the Golgi apparatus, or the Golgi bodies, whichever one you want to call it. Either way, if it's coming from the smooth or the rough endoplasmic reticulum, you create a little vesicle. And again, it's basically just a piece of the membrane that make up these organelles. Because remember, every organelle is membrane bound, including your endoplasmic reticulum and your Golgi bodies and your nucleus. Everything is membrane bound. Nothing is just sort of hanging out. So once you package it in a vesicle, it has to go somewhere, and it goes via transportation on your cytoskeleton. So basically hops on a little rail line, which is your cytoskeleton, and transports through your cell to the Golgi apparatus. Now, once it makes it to the Golgi apparatus, you should remember that the Golgi is essentially the USPS of the cell. It packages. Essentially, what ends up happening is this little vesicle fuses with the Golgi apparatus. It moves from one side of the Golgi to the other side of the Golgi. Just essentially, this vesicle just binds, fuses, escapes, binds, fuses, escapes. It sort of just punches its way through the Golgi, but it's on its way through the Golgi. Essentially, it gets tagged, and so if you make a protein that, say, you want to export from your cell, well, it gets tagged with the molecule here, and once it's tagged in the outside of this membrane, it sort of reaches the outside of this membrane here. It then has a tag on it, and now it can be sent to wherever it wants to go. But again, once it sort of leaves a organelle here, it hops back on the cytoskeleton and it goes to its destination. But again, it's tagged to say, I'm going to go here. Like, I'm going to go back to the nucleus. I'm going to go to the mitochondria. I'm going to go to the lysosome. Or if I'm going to go to the outer membrane. But again, it's all packaged in these little things. Nothing is sort of hanging out in the open here. Everything is always packaged inside of a little membrane. And so most commonly, well, not most commonly, but commonly, you get something that comes out the back end of your Golgi apparatus here, and it will then go bind to the plasma membrane. It literally just fuses to any other membrane, and it opens up, dumping its contents into where it needs to go. And this is the endomembrane system. This is how everything works. So once you make that protein, or you make that lipid, or you make that compound you need to make, it gets packaged on a little vesicle. And again, the vesicles are just parts of the membranes of these different organelles. It's packaged in one of these vesicles, hops on the cytoskeleton, and you'll see sort of how it moves on the cytoskeleton in a moment. And then it heads to the Golgi. Once it heads to the Golgi, it's tagged and sent to its final destination. So that's the idea of the endomembrane system. Really simple, but really, really effective at doing things fast. I know this sort of process seems like maybe it's a lot of work, but remember, this is something that happens incredibly fast, right? Like incredibly, incredibly fast. You know, for instance, in your liver, you do thousands upon thousands of these sort of reactions every second across your entire liver. So this means you can do these within, essentially a few seconds. It's not something that takes a long time to respond to. So if you needed a protein right away, you could do this, you could transcribe it, get it to the ER, and package it and sent somewhere within a few minutes. So you can respond very fast. I know this seems like maybe it doesn't seem complicated to you, but it can seem like a complicated sort of system, but it's something that can act really, really quickly, which is an important thing because, at the cellular level, you need to be able to respond rapidly. Think about listening. Think about me talking, right? I have to be able to do this rapidly on command, right? If I want to snap my fingers, right, I can do that on command. And all that, again, is all facilitated by this system. which I think is really cool. It's very simple, but it's something that can do it quickly, and it can be very adapted depending on what you need to do. But the sort of the next thing is how do we know where things need to go? And the idea is we have to tag them. And so proteins and lipids have to get a zip code attached to the end of them. And this basically tells them where to go. So you can think of the zip code of like, I live in the town of Easton. So pretend like I need to send something to Easton. I tack on 02375, and then the post office knows it's going to Easton. So that's the idea here. So when we're thinking about this, we have what's called the signal hypothesis. So this is it's a pretty well supported hypothesis, but the idea being we have our ribosomes and our messenger RNA here, and we have the cytoplasm here, and we have the endoplasmic reticulum here. The idea being, if we have a ribosome and we are making a protein from an mRNA, we have what's tacked on to the end, what's called a signal sequence. Every time you're producing a protein, your cells will intuitively attack what's called a signal recognition particle, or an SRP, to it. Now, once you're sort of making this protein, you attach this signal recognition particle. Well, what does that exactly mean? Well, the particle itself, it just basically is a protein, or some sort of carbohydrate. It depends. And what this does is it interfaces with a channel on a membrane of an organelle. So the idea being is that a signal recognition particle is specific to each organelle. So if you have a signal recognition particle for an endoplasmic reticulum, it can only bind with a protein on the membrane of the endoplasmic reticulum. It won't be able to bind to a lysosome. It won't be able to bind to your nucleus. It will only be able to bind to your endoplasmic reticulum. And that's it. And so the idea being is, as you're synthesizing this, you tack on one of these particles to know where it's going to go. So in this case, it's going to the endoplasmic reticulum. It's essentially this recognition particle will bind to a protein that's embedded in the membrane, and you'll notice is that as it binds, it's essentially shuffling the protein through another protein. So a mature protein here. This is sort of an unmature protein. It's one of those channel proteins. So it's one of those transmembrane proteins we talked about a few classes ago, or actually last class. And essentially just spits the protein straight through that protein into the space of the endoplasmic reticulum. And once it gets there, it's inside there, and it can be essentially changed further as it needs to be. But the idea, you know where to go by sticking one of these signal recognition particles. Again, they're mostly proteins, but they can also be sugars depending on what they are. But again, they're just recognizing the outer receptors, because it's not like you have something like they can see, right? Your cells can't see like, oh, this is the endoplasmic reticulum. All sort of sensing, all sort of recepting, all sort of, hey, I know where I am or where I'm going to go, is all done by either proteins on the outer ends or sugars on the outer ends. That's how your body and your cells know what is what. So, but as you all know, nothing exists in sort of solitary. Not even bacteria live on the desk where I'm sitting. Everything exists together. And in particular, when we're thinking about animals, we're thinking about things existing in organs, right? Your liver cells, your brain cells, they all exist in the same space. They all exist and perform a very similar function, or the identical function, and they have to work together. And if your liver is going to work, you need all of your liver cells working in tandem to one another, talking to one another, to physically make your liver work. If half your liver is deciding it wants to be a kidney, and the other half of your liver is deciding to be a liver, well, your liver is not going to function very well. So your liver cells need to physically bind together, and they physically need to be able to talk in real time and perform similar functions in coordination. That's an important thing. You need to be able to coordinate your functions of cells. More importantly, you also need to be able to coordinate functions across great distances. And that great distance I'm talking about is your body. And if you think about sort of the scale we're talking about here, right? Your average human cell is about 100 micrometers, or 0.1 millimeters. Your average human, we'll say, is 1.5 meters, right? Right, five and a half feet or so, right? That's a huge distance when the sort of the building blocks of you, cells, are tens of thousands of times smaller. So sort of being able to communicate over that long distance is a big deal. But your cells do it, right? For you to for me to talk to you guys, I have to communicate with my brain to my lungs to my all the muscles in my throat and my jaw, right, to produce all these sounds and movements. It's all coordination of, again, a very long distance when you think about everything is happening at the cellular level, right? I think that's one of the really powerful things about being a very large animal, right? Think about something like a blue whale, right? It's 100 feet long. Its brain is at the front, and its tail is at the end, right? It has to send a signal 100 feet to say move your tail to swim, right? And that's a huge distance when you're thinking about things at the microscopic level. But but for organs to work, they have to talk, and for your body to work, it has to talk. Sometimes short distances, sometimes long distances. And so when we're thinking about how things are connected, there's three ways your cells connect together that they join. And so there's three ways. They're tight junctions, desmosomes, and gap junctions. They all have very specific functions. So tight junctions essentially form watertight seals. You could think of it, this example gives you caulking around a tub, but you can also think about it like gluing two pieces of paper together. Basically just really tight gaps that allow your cells to basically stay together. Now, this is what happens in your skin. And so your skin, as you know, is pretty much waterproof. It will absorb some water, but it's more or less waterproof. And so what binds your skin cells together are these tight junctions. Really tight sort of joining together. It's all done by proteins, as you can sort of see. There's this like network here. It's all proteins. And they create this watertight seal. And this is what your skin cells are held together by. Next up, we have desmosomes. These essentially are like Velcro, and it actually does look like this. If this is a more if you this is a bigger picture, you could sort of see these little Velcro-like things. They're just proteins, but you could see they're actually physically linked together. And these are essentially what Velcro things together. They basically hold your cells together. This is how your organs are held together. These desmosomes, these Velcro-like things that fasten your cells together. And they allow your organs to stay together, but they don't create like this watertight seal in between them. It allows greater talking in between. And then finally, we have our gap junctions, which are essentially pores. They're proteins, as you can see, that sort of transmembrane protein. Oops. And they essentially act as little channels so that cells that are right next to each other can directly communicate with one another without having to sort of go in between them, right? They don't have to go in these gaps, right? So the idea being is that if you have a bunch of liver cells, they would be joined together by desmosomes, and they would essentially have a bunch of these gap junctions to send things back and forth between liver cells. In addition, tight junctions will essentially bind together the outer layers of many organs to keep them sort of tightly sealed as well. This is how organs are essentially kept together, and how they're able to communicate in real time. Because if you had cells that were sealed together by, say, a tight junction here, you wouldn't be able to directly communicate. But having this gap junctions allows us to essentially communicate in real time. If I have this cell here and it's responding to, say, because I just drank a bunch of beer and there's a bunch of alcohol in here trying to detoxify, well, it could then pump that alcohol into the neighboring cells to say, hey, help me out with all this alcohol that I just drank. So it allows real-time communication. So that's at the local level. These are the way cells are bound together locally. Now, in terms of at the body level, your cells are tagged, and I've mentioned this multiple times. Your liver cells are distinct from your pancreas and your heart and your feet and all your other different types of organs. Everything is distinct, and they have different orientations of organelles that allow them to perform their specialized functions. Now, what the idea being is they're all tagged. Every cell is tagged with its own very specific type of carbohydrate. And this tells you like, hey, I'm a liver cell. Hey, I'm a muscle cell. Hey, I'm your heart. So everything is tagged. So if something, say, makes it out into your bloodstream, and you have, say, a signaling molecule like a hormone that you need to, say, act in your heart, right? It's produced in your, I don't know, your liver, and it needs to go to your heart. Well, a hormone, say, would be produced, it'd be put into your bloodstream, and it would sort of travel through your bloodstream till it reached where it needed to go, and then it would know to bind to a heart cell because of the outside, essentially, these carbohydrate chains on your heart cells. And so this sort of keeps your organs connected but very separated. And remember, your membranes are pretty good at keeping most things from going in. Gases, water, they move freely, but proteins, other large molecules, they need to be able to they need to be transported across this membrane. And the only way they'll be able to get transported across this membrane is if they can dock with one of these carbohydrates and then be brought in to sort of the location you need them by one of these proteins. But again, they wouldn't know where to go if things weren't tagged. And that helps keep your cells distinct, right? It knows that your body knows that this is a liver cell, this is a heart cell, and so forth. But in terms of thinking about movement inside the cell, long distance movement inside your cell, in your body is done by your vascular system. So not much to talk about there. Pumping of blood is pretty straightforward. But in terms of movement inside your cell, this is what we're thinking about as the cytoskeleton. As I mentioned, everything is anchored to the cytoskeleton, and if things move on your cytoskeleton, if they move inside your cell, they have to be done inside that cytoskeleton. And the cytoskeleton is a really interesting sort of overall organelle. It's essentially, you think about like concrete poured over rebar. So your cytoskeleton essentially acts as a rebar, and your cytoplasm is essentially the concrete. But the concrete as in like liquid concrete, not as in hard concrete. Remember, your organelles are bound to this cytoplasm, which you would think about the rebar here. And the way things move on this cytoskeleton is through what are called chaperone proteins. Anyway, and then one other thing before we sort of talk about the chaperone proteins, I just want to mention bacteria also have cytoskeletons. Archaea also have cytoskeletons. They are just very different than ours. So most of them either are rings. So this is a spherical bacteria. It has one circular ring. And then our bacteria that are rod-shaped or crescent-shaped, they have essentially like a corkscrew around them. Protein, very much like our cytoskeleton, but not quite as complex as what you see here, just as a note. But in terms of movement on the cytoskeleton, is all done by these chaperone proteins. And normally I like to show these videos. I recommend taking a peek at them to sort of illustrate what's going on. So this is what a chaperone protein looks like. There's types of them called kinesins. This is what a kinesin looks like. It has a tail, a stalk, and a head. This is the basic structure. You'll notice this is very small. So this electron micrograph here is 5 nanometers. So it's very, 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 very tiny. Remember, a bacteria is 1 micrometer. 1 nanometer is 1,000 times smaller than a micrometer. So we're talking about teeny, 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 tiny levels here. Very, very small. But it has a tail, it has a stalk, and a head. And the idea being is if we have a microtubule, so part of our cytoskeleton, these kinesins actually walk along the microtubule tract. So the idea being is that the tail of the kinesin or the chaperone protein will bind to the vesicle. And remember, everything is moved in between organelles by vesicle. Nothing sort of just goes through the cytoplasm willy-nilly. So we have the tail that physically binds to the vesicle. And basically what it does, these heads will bind to the cytoskeleton. And the cytoskeletons do have directionality. They have a plus and minus end. So when you have one of these guys walking along a cytoskeleton, it will move only from the minus end to the plus end. So thinking about like reading a book, right? In sort of standard English, you read a book from left to right. And that's the same idea here, right? You walk along the microtubule only from minus to plus. But the idea being is that these actually physically walk. And it like I said, take a peek at these YouTube videos, seeing it walk. It basically, they're like little legs, and it essentially walks along these tubules. And remember, if we're going to do this kinetic energy, right, we need ATP, right? You can't this just won't happen spontaneously. You need energy, and remember, ATP is the way you get that energy, right? So cellular respiration to make ATP. But again, watch the videos. It literally walks. It looks like a dopey thing walking, but essentially you walk things across your cytoskeleton, and it's all powered by ATP, which I think is cool. I love watching these videos. One of the things I think is sad about not being in-person class or having in-person classes is that we don't get to watch all these funny videos together, because I like these videos personally. But that's the idea. It walks things across your cell. So if you're trying to go from the smooth endoplasmic reticulum to say a lysosome, you grab one of these chaperone proteins, it attaches onto your vesicle, which has whatever you want to get rid of in it, and it walks it. It looks like Gumby, and it walks all along these things to get to its final spot. In addition, your cytoskeleton has the capacity to move your cells, and so cells aren't stationary. You can do one of two, and so your cells aren't stationary, and they can move. And this is sort of not really something we think about for sort of human cells, right? Your liver cells aren't moving, right? Your liver is anchored, right? Inside your liver, your cells aren't moving. Your blood cells are moving, but those are all moving because they're being pumped. But there are special types of human cells, in particular sperm cells, that can move. And there's all sorts of microscopic eukaryotic organisms that actually can move. And the sort of the ancestor to all eukaryotes likely also had the capacity to move as well. When we're thinking about how eukaryotes move, or even how bacteria move, there's two ways. You flex your cytoskeleton, or you move with a flagella or some other structure that resembles a flagella. And so this is what we're talking about here. So you can move by one of two ways, with the physical structure. And this is so we have cilia. So this is a protozoa. It's a microscopic eukaryotic organism. And for this protozoa to move, it essentially it uses ATP to essentially vibrate these little structures. They're little hairs. They're called cilia. And it essentially powers this organism through a liquid. So that's cilia. It's a nice little hairy protozoa here. And then we have what's called the flagella, which is essentially a whip-like tail. And so we have just a microscopic eukaryote here. And what it does is essentially rotates this flagella or flagellum, you can call it either way, and essentially like a corkscrew fashion. It rotates it over and over again to power itself physically through a liquid. Because remember, everything exists within liquid. As I mentioned, prokaryotes also have this, or our bacteria have it. They have these flagella that are embedded in their membranes. And I would recommend, if you have a spare moment, to watch what this looks like, how bacteria physically move in real time by essentially spinning this whip-like flagella. They can move pretty fast. So the fastest swimming bacteria can swim 700 micrometers per second. And that's may not seem like it's very fast, right? Because a micrometer is very, very small. But remember, that's something moving 750 times the length of its body every second. So that would basically be like me. I'm 6 feet tall. That'd basically like me moving 1,300 feet per second, which is incredibly fast. So flagella can power bacteria to go really fast. And I would recommend taking a peek at this YouTube video because I think it's really fascinating to see how they physically move. But again, it's all powered by these whip-like tails called flagella. And it's not like a fin where you sort of wave it back and forth. Again, it's like a spinning corkscrew that powers them through liquid. And that liquid, again, is water. Our eukaryotes move as well. So we have for the males in the class, you have your sperm. It is powered by a flagella. The flagella is very similar to an archaeal flagella. So this is a bacterial one. Archaea have a very different flagella, and their flagella actually resembles our sperm flagella. But again, your sperm, if you're trying to, you're talking about sexual reproduction, that sperm is making its way to the egg, powered by a flagella. And so if you want to see what that looks like in real time, not the fertilization part, but just what a sperm looks like when it swims. Take a peek at that. We also have amoeba, which are pretty cool. They're microscopic eukaryotes, and they move by essentially like gliding. They stretch their cytoskeleton out, and they pull themselves along. So you can think of like a slug. It stretches its little front part out and pulls its back part. It stretches its front and pulls its back. Again, that's all done by stretching its cytoskeleton. And so if you want to sort of think about this at a whole organ level, here's a nice little YouTube video that sort of summarizes it. Anatomy is sort of a more specialized thing that we're not going to really spend a ton of time talking about anatomy. But if you want to know, you want to sort of do it like a 10-minute dive into what sort of how your organs are linked together and how things work. This is a nice little YouTube video that I'd recommend taking a peek. But so that's cells. And remember, we're thinking sort of about how cells work, and that's the sort of the functional part of cells. But we've just been talking about individual cells. We've mentioned organs and tissues and stuff like that. But when we're thinking about things, we haven't really discussed how we go from a fertilized egg to a 30 trillion mound of cells that makes a human. How do we go from a single cell to multicellularity? And that's what we're going to spend the rest of the class talking about, how we go from one cell to two cells, two to four, and so on and so forth. And how do we make our sex cells? How do we make sperm? How do we make egg? It all starts off, though, again, back to this DNA. We got to think about the central dogma of biology, how information is propagated from storage to action. Because for you to make more cells, for you to make a sperm, for you to make an egg, everything needs DNA. Everything needs DNA. And so just as a refresher, all the DNA that makes up your genome is all the DNA that makes up you is housed in what's called your genome. And your genome as a human is comprised of 23 chromosomes, and on those chromosomes, 23 pairs of chromosomes, 46 total. And your chromosomes have what are called genes on it, and these genes are what you use to make mRNA, which in turn makes protein. Just as a reminder, 23 pairs, so you have 2 copies of every chromosome. Those pairs are called homologous chromosomes. And you have homologous chromosomes, homo meaning the same, so they're identical to one another. So this could be chromosome number 1, and this would also be an identical copy of chromosome 1. And because they're essentially identical, they have copies of the same genes. So if this is a gene that makes a protein for your hair, you would have 2 copies in your genome. You get one of those copies of those chromosomes from mom and the other one from dad. Right? Makes sense. Half your genome's from your mom, half your genome's from your dad. And remember, those genes, they can be similar, they can be identical, or they can be very different, but variations in a gene are called alleles. So we'll just do a quick little recap of how prokaryotes divide, because again, we're talking about division. We're trying to talk about how you get genome from essentially 1 cell to 2 cell. And so prokaryotes, again, they were produced by this process of binary fission where 1 cell becomes 2. And this is a video if you want to take a peek at what this looks like in real time. It's pretty fast. Remember, as we talked about last week, essentially a bacteria can divide every 20 minutes or so. So they reproduce really fast. But in terms of binary fission for bacteria, we have the bacteria. It has its chromosome. The first thing it does is it makes a copy of its chromosome. So it has 2 identical copies. And then essentially it buds and forms 2 daughter cells. And so these 2 daughter cells are genetically identical, provided the bacteria didn't make an error replicating its genome. So that's how bacteria reproduce. Very, very simple. Nothing really involved. It happens fast. But again, not very involved. Make a copy, split things off, go from there. And that's just why it can happen fast. Very, very simple, very, very small genomes, so it happens really quickly. Now, that's bacteria, but your body is different. Animals are different. Plants are different. And your cells are divided into 2 distinct types of cells. So we have non-reproductive cells, which are called your somatic cells. This is everything except for sperm and egg. That's what a somatic cell is. Everything in your body that is essentially not housed in your genitalia, and by your genitalia I mean your ovaries or your testicles. Anything that's not housed in there is a somatic cell. Anything that is housed in there, that's either a sperm, an egg, or if you're a plant or a fungus, that's called a spore. Those are your gametes, or your sex cells. And so we've sort of been talking about how you have 2 copies of every chromosome. And that's true. All your somatic cells have 2 copies of every chromosome. And so they are what are called diploid cells, meaning they have 2 copies of each chromosome. However, your gametes, or if you're a plant or a fungus, your spores, are actually haploid, meaning you only have 1 copy of each cell. 1 copy of each chromosome. And so these are the 2 distinct types, but they are linked together in some shape or form. So the idea being, we have sexual reproduction. So between our male and our female here, they produce through a process called meiosis. And we're going to talk about this today, either an egg or a sperm. And each of these gametes has only 23 chromosomes. So they have half the number of chromosomes typically found in your average cell, which makes them haploid. But as I'm sure you all know, making a baby requires a sperm to fertilize an egg. And once you fertilize that egg, again, it's fertilization, you produce what's called a diploid cell, meaning that fertilized egg, which is also called a zygote, now has 23 pairs of chromosomes, or it's diploid. Now, once you fertilize this egg, You then go through a process called mitosis to make essentially more copies of this cell. And that's how you tie the birds and the bees together with essentially making a larger individual. The original production of gametes, which again come together to fertilize, is done through a process of meiosis. And once meiosis is done and you have a fertilized egg, mitosis takes over, taking the single cell and making it into the trillion cell person that you are. But remember, we all started from a single fertilized egg that went through tons and tons of mitosis events to make the people that we are today. So let's talk about eukaryotic cell division. So again, this is called mitosis, and this is the way we make new cells. So if I'm not like I would be shaving, but if I was shaving like this guy here and I cut my cells, well, I don't just leave an open wound, right? I don't just leave open wounds on my body, right? I have to repair those wounds. And if I'm going to repair those wounds, well, I need my cells that are surrounding those wounds to undergo mitosis. So again, cellular division to make more cells to repair my wounds. Makes sense, right? If I have a wound in my skin, I need to fill it with new cells. And I do that by mitosis. Now, conversely, if I need to make new cells because I'm trying to grow, so in the case here we have a plant root or a plant stem trying to grow, well, I need mitosis to take my existing plant cells to make copies of them so that I can physically grow. And so mitosis does both things. It takes small numbers of cells and makes large numbers of cells, or mitosis can repair things. And so mitosis is involved what we call the eukaryotic cell cycle. And it's pretty straightforward. So the idea being is we have sort of three distinct cycles here. So we have first, which is called interphase. And interphase is essentially divided into three subgroups. So we have what's called gap 1, we have DNA synthesis, and gap 2. And so when your cells aren't trying to divide, they exist in gap 1 phase. And so gap 1 phase is essentially just, I'm doing my normal thing. Like my skin is doing its thing, my liver is doing its thing. They're just essentially in the normal state. They're trying to help your body do its thing. So that's gap 1 phase. In addition, after gap 1 phase, we have DNA synthesis, where you essentially start to get ready to divide. And the first step in doing this is replicating your entire genome. And as I mentioned, bacteria do this very quickly. It takes about 24 hours for your average human cell to replicate your entire genome. I don't think I've given you a number on the total number of DNA bases in your genome, but it's about 3 billion. So it takes a very long time to make a copy of something that is 3 billion DNA bases long. So that's the first thing. It takes about 24. I'm sorry, that's the second thing. It takes about 24 hours to do. Once you make a copy of your DNA, you enter what's called the gap phase, and you continue to grow, and you essentially start to stockpile nutrients. You start to make copies of your organelles that essentially making you ready for mitosis. So once you're just kind of doing your normal thing in gap phase 1, you synthesize your DNA, and then you prepare for cellular division. You make copies of your organelles, because when you make a daughter cell, that daughter cell needs to have everything it needs to survive, which includes organelles. You also make sure you have enough nutrients and as well as ATP to do this process. Because as you can sort of see from down here, you're going to take one cell and split it into 2. So you need a lot of energy to do that. You need a lot of materials to do that. So you have to prepare for that, and that's what gap phase 2 is for. Once you exit gap phase 2, that's when you enter mitosis. And this is essentially when the cell essentially is doing all these sort of different steps that we'll go through in a second to essentially separate one cell and make it into 2. And then finally, you have what's called cytokinesis, which is the actual physical separation of the cells from one another. So you have this 5-step process. Again, this is how the vast majority of your cells spend their time. It's in gap phase 1. Then when you're getting ready to divide, you start to synthesize your DNA. You start to replicate your organelles, get all your energy in order, and then you physically undergo mitosis, which is then followed by cytokinesis, which is essentially just the physical separation. But let's talk about mitosis. What does this look like? And so if we have a sort of a simplified version of our mitosis, it has multiple steps to it. And these steps are listed above. There's a few in between here, but we're not going to talk about those. This is just a very simplified thing. So the idea being is you have a normal cell. If it's just growing normally, it's an interphase, remember? So that sort of interphase is this part where it's getting ready to divide or it's just normally growing. Once it makes a copy of the genome and it's ready to divide, it then enters mitosis, which starts with metaphase. And then there's a few different steps along the way. But one of the most important things that happens in metaphase, in mitosis, is all your chromosomes will actually line up on the center part of your cell. So all 23 pairs will line up in the direct center of your cell. And what ends up happening is you have your centrioles, so those little organelles I told you about earlier in class that are only found in animal cells that I told you I'd come back to later. Those centrioles will essentially migrate to the poles of your cells, and they'll extend out these little spindle fibers. And these little spindle fibers are essentially like your cytoskeleton. They're essentially like little tubules of proteins that they extend out. And these centrioles, they'll essentially extend out these spindles, they'll attach to your chromosomes, and essentially grab them and pull them apart. And the idea being is that half your chromosomes, so remember to get to this point, we had to make a complete copy of our genome. One full copy of the genome will migrate to one side, the other full copy of the genome will migrate to the other side. And once everything is sort of separated on these two poles, cytokinesis will occur. Again, that's separating, producing two daughter cells that are identical. They're identical because they have the same genome. They're also identical because they have essentially all the same organelles as well. So, the original cell, it's mitochondria. Half of them go to one side, half of them go to the other. It splits its endoplasmic reticulum in half. Half goes to one side, half goes to the other. And does this for all the organelles. It splits them in between the two daughter cells so that the daughter cells, after cytokinesis has occurred, they have everything they need to just start becoming a cell. They can start to do whatever they need to do. So if this was in your liver cell, these daughter cells that we produce have everything that they need to start being a liver cell. Because you can imagine, like, your body kind of can't skip a beat, right? Your liver can't skip a beat. Your heart can't skip a beat, right? It needs to keep going. And so these daughter cells need to have everything they need to start working right away. Because your cells really never stop. That's sort of the unfortunate reality of it. But this is mitosis. This is the process. Copying your genome, making copies of all the things you need, getting all the nutrients and energy you need, and then physically separating the DNA and the organelles through these spindles, and then the physical division part. This is cytokinesis. And again, this is what creates duplicate cells. And this is just a graphical version of what we just talked about. Again, interphase, we're making the DNA and all of that stuff, getting ready. The chromosomes come together, they line up, they get pulled apart, and then this is what happens. They divide by cytokinesis. So this is mitosis. And once you were essentially a single cell, once you were a fertilized egg inside the womb of your mother, mitosis took over. So we are the 30 trillion cell mass that we are because of mitosis. So we took one cell, and that one cell started undergoing mitosis. So that one cell became two cells, and that two became four. Four became six, and so on and so forth exponentially over the period of, essentially up until we're 18 years old, or when we stop growing, continuous mitosis events over and over again to make us that 30 trillion cell mass that each one of us is. So that's mitosis. Again, produces two identical daughter cells. This is how you started off from a fertilized egg and it became the massive cells that you are. But this is also how you physically repair a cell or some damage inside your body. So that's mitosis. Super important, not particularly very complicated, but again, super important for eukaryotes to do. The cousin to mitosis, which is happening in meiosis, is basically how you make sperm and egg. So this is a process that occurs in your gonads, which is, again, women would be your ovaries, men would be testicles. And this is meiosis is how we make our sexual cells, and these sexual cells are called gametes. And all meiosis really is essentially 2 rounds of mitosis without any DNA replication in between. So the idea being is, if we were thinking about mitosis, right, we have interphase. We have interphase where we make a copy of our genome and all the organelles we need. And then we have that first section of mitosis where we make 2 identical daughter cells. Well, in meiosis, that exact same thing happens. But what ends up happening is right after meiosis 1 finishes, that's something that's very identical to mitosis, we have another division right away. So we don't have any genome replication occurring here. So that what we get is 4 daughter cells that essentially have half the number of DNA that they're supposed to, because again, we have no DNA replication that occurs here after meiosis 1. So the idea being is we have interphase. Again, that's when all the things happen, making copies of chromosomes, making copies of organelles, getting the energy we need. Then we undergo meiosis 1, which again is essentially identical to mitosis. We have the alignment at the center, we have the pulling apart, and then we have cytokinesis, making 2 identical daughter cells. Meiosis 2, almost the same thing, just no replication of DNA. We have, again, they line up at the center, they get pulled to the side, and then we have essentially separation, producing, again, 4 identical daughter cells. But these daughter cells only have half the number of DNA. Because remember, the idea being to make these cells, we're trying to do sexual reproduction. We're trying to combine DNA from one parent and the another together so that the offspring only has 1 full genome. So that's meiosis. It's almost identical to mitosis. It just, in between meiosis 1 and meiosis 2, there's no replication of the DNA. And just as a sort of a final note, women do things slightly differently. Males, we always produce 4 sperm cells every time. And our sperm cells are cheap. They're easy to make. We produce them constantly, but women do things differently. And this is for a very specific reason. So women do things differently because an egg is, as you can see here, is significantly larger than a sperm. And this is the case across pretty much every animal that we know of on the planet. Eggs are huge relative to sperm, and I think that's pretty well illustrated here. And the reason being is that sperm, they need to move, right? So they need to be agile, they need to swim, so on and so forth. Eggs essentially provide all the nutrients that the developing, essentially infant, the little ball of cells inside of any given animal, needs to survive. So there's a huge investment that women make relative to men. So sperm is cheap, it's easy to make, who cares? But to make an egg, it requires a much more heavily invested form of meiosis. So the idea being is, if we're trying to make eggs, the idea being is we have essentially we start to do meiosis 1. And the idea being is we actually have meiosis 1 produces 2 distinctly differently sized daughter cells. So one is pretty large, and the other one's kind of dinky. And that little dinky cell actually ends up going on to essentially be recycled. It essentially dies. It's just very, very tiny. So you have one very, very large cell. Then we have, again, another replication event of this very large cell, but again, because this little one has died off. We have another replication event. Again, we have one very, very large egg and one very tiny one, and this very tiny one then essentially goes on to die and be recycled. And what we're left with is a single, very large egg. That's our gamete. Again, it has half the amount of DNA, but it has all the things that we need. It has a huge store of energy. It has all the organelles we need. It's huge, again, relative to your average sort of gamete out there. But women put a huge amount of investment in this line so that essentially whatever they make from this, the offspring, has all the things it needs to survive. And in terms of production over time, for women, this is done essentially all at once. So you do this meiosis process inside of your ovaries, basically when you're developing in the womb. You produce all these eggs. You produce about 1 million of them when you're developing, and then about, 60% of them die or so. So you basically, once you're born and once you sort of hit your teenage years, you have all the eggs you're ever going to need inside your ovaries. Males, we do things differently. We produce gametes essentially constantly because that's just what we do. So we produce new sperm every day and replace it every 7 days. So that's sort of the difference between men and women in terms of production of gametes, at least for humans. And so with that, that's going to end our talk today about cells. And so now we know how cells work. Now we know how cells divide. Now we know how sex works. And so we're going to dive a bit deeper into genetics and things like that in the coming classes, as well as thinking about how plants and animals have developed and how we've grown and how we've diversified. So with that,